one from paternal parent, okay? So in here, we have multiple genes, okay, that has different information, hair colors, eye color, disease, so many different information, okay? Then, out of these, we talk about one gene, okay? One gene, all right? Then, we call these to each alleles, okay? Then, out of these allele, all right, we have dominant allele and recessive allele, okay? Or it could be identical. It could be dominant, or recessive, okay? But for example, if we have heterozygous like this, okay, then this is dominant allele, dominant, okay? And this is recessive allele, okay? And when you're talking about the phenotype, okay, if you are talking about the phenotype, then always whatever that's the dominant allele only get expressed, okay? So dominant allele, yeah, is always what gets expressed, okay? So that was the law of dominance, all right? And then we also talked about the law of segregation, right? So when these two, okay, this is contained in one parent, right? So when these two actually go through meiosis, okay, then the gamete that gets produced is actually haploid, right? So it actually contains one information of the homologous pair, all right? So these two actually get segregated into different gametes like this, okay? And this also fertilized with the random gamete from another parent. Okay, so that's the two laws that you have to know. Also, we talked about monohybrid cross, which is a cross between just one allele. Okay, we don't focus on any other allele, but just one allele that has specific information, like a height or color of flower, stuff like that. Okay, so today we're going to do another law. So that's on page 117, law of independent assortment. Okay, so if you read this, law of independent assortment applies when a cross is carried out between two individuals that are hybrid for two traits on separate chromosomes. Okay, so when you go to the next page, this is what it's talking about. So now, if you're talking about two traits, okay, not one, but two, okay? All right, so if you have a parent cell that actually contains two chromosomes, okay? So out of two chromosomes, okay? So during meiosis 1, this is meiosis 1, okay? Meiosis 1, metaphase, then we focus on two chromosomes only out of 23 pairs of chromosomes. Then here on the first chromosome, we only focus on one allele, like this, okay? And also on this chromosome, we also focus on one allele like this, okay? Then, during the metaphase, right, actually the microtubules, okay, the spindle fiber actually gets attached to the chromosome and then pulls these to the opposite ends, okay? But when this gets pulled together, all right, these two can actually go to this side, all right? So this can be large A, large T, or and small a, small t, like this, okay? But also, this can be switched like this, okay? How this is going to be aligned is random, all right? So this can be this, okay? So instead of this, also, this can be small a, large a, like this, okay? Then, if these two get actually separated to the opposite end, also, this gametes can be small a, large t, okay? And this can be large a, small t, all right? So, out of this, actually 4 different types of gametes can be produced, okay? So this is what this is talking about on page 118, all right? So if you look at the figure 9.2, okay, so this is the point where it gets segregated, all right? So right below figure 9.2, it says the only factor that determines how these alleles are inherited is how the homologous pairs happen to line up in metaphase of meiosis 1, which is a random event, okay? So this can be actually large T, large Y, and then small t, small y, or these two, okay? Then actually, let's go with y, okay? So if you are crossing, okay, this parent with